Okay, good evening, everyone. Again, welcome to Ontario Nature's Ontario Reptile and Amphibian publication, celebrating over 10 years of community science. We're really excited to have this event this evening. I'm just going to do a little bit of housekeeping before I turn it over to my colleagues. So as you should have heard when you entered the webinar, it is being recorded, and your faces and names will not appear on that recording. And the recording will be available later for you to view again and to share. So you'll get a follow-up email in a couple of days after this webinar that will have that recording contained within it. So that will be available to you. If you have any questions for the panelists, we will have some time for questions at the end, not a whole lot, but some. So if you would like to ask those, we'll do that at the end, and you can put those in the Q&A, and we will moderate a Q&A session at the end. If you have other things that you'd like to discuss, or comments or technical issues, you can put those in the chat. Okay, so please use the Q&A for questions for the panelists and the chat for technical issues or any other problems like that. Thank you very much for joining us. And with that, I will turn it over to Jenna Quinn.
Truly collaborative community science project, the Ontario Reptile and Amphibian Atlas. I'd like to start by recognizing that I joined today from my home in Kitchener, which is part of the Haldimand Tract, and on the traditional territory of the Neutral, Anishinaabe, and Haudenosaunee peoples. I offer my gratitude to the deep traditional knowledge and Indigenous stewards who have cared for this land since time immemorial, and to the many diverse First Nations, Inuit, and Métis people who now call this land home. We are all treaty people with a responsibility to honor all our relations and be active in the calls for truth and reconciliation. I also want to acknowledge that relationships with and observations of reptiles and amphibians existed long before written records. And so with that in mind, it's important to note that this project that we're discussing today offers a culturally specific perspective about our understanding of reptiles and amphibians in the province. And historical and ongoing Indigenous stewardship of these lands has been essential to the conservation of biodiversity across Turtle Island. The Atlas is a huge effort, including observations from more than 12,000 contributors, and I hope many of you who are joining us tonight are those contributors. And so to get a sense of our audience here, we're just going to launch a quick poll, and please let us know how you've been involved with this project to date. So you should shortly see a poll up here. So you can choose an answer there that best represents you. And if you're just learning of this project for the first time, then thank you very much for being here. I hope you walk away tonight with some new information and some new resources to inform your conservation work or your environmental passions moving forward. And we'll just give people a few minutes or a few seconds more to answer. So while we wait for everyone to fill out the poll, I'd just like to give a special thank you to the regional coordinators, the organizational champions, the Atlas Steering Committee, and the Publication Steering Committee, who were really essential to not only support the Atlas during the data collection phase, but also the many people who were authors, reviewers, and more during the preparation of this publication. We'll give it maybe 10 more seconds. Okay, perfect. So you can see the results of the poll on your screen. So we've got lots of newbies here tonight, which is great. Very happy to have you and to have you a part of the Atlas journey here. And so you can close the poll box, I believe, on your own on your own screen once you're done looking at it. And then we can move forward. So yeah, thanks for being here, and thanks for everybody who was a part of the project in the past, and hopefully many of you here will continue to be a part of the project moving forward. So atlases are projects that collect observations of a target species group from many sources in order to improve our knowledge of the distribution of those species. So you may be familiar with the Breeding Bird Atlas, for example, but there's many examples of atlas projects around the world that cover diverse taxa. So what happens is the province got split into 10 by 10 kilometer square grids, and observations of reptiles and amphibians were then collected from a wide range of individuals and organizations, with each observation being associated with one of those specific grid squares. And data was submitted from lots of different sources and then verified and vetted by a collection of staff and volunteers. So over the lifetime of the atlas, we have a cumulative database of nearly 500,000 observations, over 250,000 of which were collected through the 10 years of the ORAA. The success of the atlas has largely been from the immense efforts of over 12,000 researchers, naturalists, and members of the public and other collaborators, including over 200 organizations across the province that all submitted data to this project. And of course, the growth of the atlas since 2009 would not have been possible without the efforts in engaging the public, with over 27,000 people participating in reptile and amphibian focused events over the last 10 years, or since 2008. And that was over 260 events that have been run in that amount of time. So lots of opportunity for engagement. So what did we see? What were the species? Well, let's find out. We're going to launch a second poll here, so you can take your guess of what species group fills the largest piece of the pie here, or in other words, which species group was observed the most during the Ontario Reptile and Amphibian Atlas. So you can choose from all the species groups that will appear on the poll, and just guess which one fills the largest piece of the pie. But while you're doing that, you can also consider what might fill the next largest pieces. So you should see a poll appear shortly. Your options are turtles, frogs and toads, snakes and lizards. It's important to note that our lone lizard is lumped in with the snakes here tonight, and salamanders and newts. Lots of guesses coming in. Give it 10 more seconds, maybe. Awesome, great. Great, okay, thanks for answering. You can see the results on your screen shortly, and you're all great students because many of you, 65%, guessed the right answer, which is frogs and toads. So again, you can close the poll box on your own screen if it's in the way, and we can fill in this pie. So yes, the largest piece here is frogs and toads. They made up about 293,000 of the observations in the atlas, followed next by snakes and lizards, which made up about 86,000 observations. 79,000 observations or thereabouts were turtles. And the last smallest piece of the pie there, salamanders and newts, about 24,000 observations. So I think that's enough from me tonight, but I'm very happy to introduce to you all an excellent panel of speakers, all of whom were deeply involved in the Atlas project. Joe Crowley was project coordinator from 2009 to 2010. Joe is a professional biologist with expertise in herpetology and species at risk conservation. He's currently a species at risk biologist with the Government of Ontario, where he's worked since 2010. James Patterson was project coordinator from 2011 to 2013. James is a research scientist at Ducks Unlimited Canada, and he is a quantitative ecologist and conservation biologist working to improve solutions and test how biodiversity responds to global change. Tanya Pulper was coordinator from 2013 to 2016. She's focused most of her career engaging the public in the conservation of species, and she's now a wildlife biologist at Environment and Climate Change Canada. And Emma Horgan was project coordinator from 2016 to 2018. She's an ecologist with over 15 years of experience in conservation biology and research, and has been extensively involved in community science initiatives She currently works for the Canadian Wildlife Service, Environment and Climate Change Canada in the Protected Areas Program. And finally, Patrick Muldowen is unable to join us live this evening as he's currently living and working in the Galapagos, overseeing the Galapagos Tortoise Movement Ecology Program, but he did provide a pre-recorded video spotlight for us tonight, highlighting some of his interesting research, largely based at the Algonquin Wildlife Research Station, where he's involved in long-term studies of salamanders and turtles. So before I turn it over, I'd just like to say that it's been my pleasure to cheerlead this project past the finish line, but truly it wouldn't have been possible without the work of these fine folks on your screen here and many others not pictured, whose passion and dedication for conservation, particularly reptiles and amphibians, is the driving force behind this project and so many others. So it's with deep gratitude that we have them here tonight. So each panelist will speak to us a little bit about a part of the Atlas project, and at the end, everyone will join me on screen and we'll have a Q&A. So as Leslie said at the start, please pop your questions into that Q&A box along the way, and we'll get to as many as we can at the end of the night. So with that being said, I'll welcome Tanya to the screen and turn it over to her to talk a little bit about the history of the Atlas project.
Hi, everyone. It's great to see so many people online today. My name is Tanya, and I was the coordinator of the Atlas and then manager from 2013 to 2019. And today I'm going to highlight the history section of the publication. And I've been told and promised that that's not because I'm the oldest panelist here. So next slide, please. So tonight I will be, during this presentation, I'll be doing a lot of name dropping. And that really is because so many of the herpetologists don't get the press that other biologists do. I'm not sure what other press other biologists get, but really these guys are not well known for lifetime of career of conservation. So I really feel like we need to give credit to those working hard in the background. So I'm going to start off with the pioneers of herpetology. And it starts off with the first herpetologist that was hired, or the first herpetologist in Canada, who was Clyde Patch. Now, Clyde was, he was at the Canadian Museum of Nature from 1918 until 1952. And really, he was here during the Depression, and not a lot of funds were there. And he was sort of deemed a really creative person to do and move things with very little money. And I think that that's really cool. And we may have seen that a lot in the NGO world as well, but probably not to the extent as Clyde during the Depression. Then we would go to E.B. Shelley Logier. Now, he was an artist and herpetologist with the Royal Ontario Museum of Nature, the ROM. And in 1955, Logier and Toner created a dot distribution map called the Snakes of Dot Distribution Map. And then he went on to publish the Snakes of Ontario, which is, I have a copy here on my desk. And it's a really great resource of species account, checklist, and distribution maps as well. Then I'm going to move to Sherman Bleakney. So I have Logier here in the bottom left corner that you can't really see. And then Dr. Bleakney is here with this huge sea turtle skeleton. And he completed the first PhD dissertation on herpetology in Canada. And then went on to publish a zoographical study of amphibians and reptiles of Eastern Canada in 1958. Now, then we have Francis Cook. So I've got two pictures of Dr. Cook here. The first one is close to the start of his career, I believe, where he started as a field assistant to Bleakney, Dr. Bleakney, and then succeeded him as the curator of and herpetologist at the Canadian Museum of Nature. He and his wife created a, he created the book Introduction to Canadian Amphibians and Reptiles with a large help from his wife, Joyce. And it was the first in-depth book on reptiles and amphibians in Canada. And all these contributions we have built on for all the Atlas events since then. But also we still use these resources today. And I have like, as I said, I've got the Book of Snakes of Ontario still on my desk and I still go to it on a regular basis. Next. So in 1984, the Breeding Bird Atlas was winding down and a group of dedicated naturalists were looking for a new project. So many taxa were discussed, and these are the variety of dedicated naturalists here. I'm not sure if people can see the names at the bottom here, but we have Mike Oldham, Marty Obard, Mary Garchor, Peter Carson, Wayne Weller, Gary Allen with a very stylish haircut of Gary, Don Sutherland, and Ross McLaughlin. And I didn't have a picture of Ross, so I put his McCulloch, sorry, I put his ROM herp guide up there as well. But they started talking about a variety of different taxa, and then Mike and Don went to Mother's Pizza afterwards and just decided for everybody that was going to be the reptiles and amphibians. And they would then start rallying interested naturalists who were looking to continue the momentum from the Breeding Bird Atlas for those contributors to submit records of reptiles and amphibians. The first call for submission was put in the Toronto Star, pictured above here, as well as mailing 200 potential participants. Outreach and promotion for the Atlas basically relied on word of mouth, as well as articles in magazines such as Seasons, which was Ontario Nature's or On Nature's predecessor to this, which was Ontario Nature's On Nature's predecessor. The dual purpose of the community science project was to engage people of varying ages and skills and improve knowledge of the distribution of these species. And this theme is something that we will see throughout the this atlas as well as future atlas projects. Next slide. So the Ontario Herpetofauna Summary was born. And this was moved from, with Mike Oldham from the Ontario Field Herpetologist Group to the Natural Heritage Information Center, or NHIC, in 1993. A lot of groundwork was put into collecting his historical records and soliciting new ones. Observations were mailed in on species data cards like the one you see here, that were basically the size of rescue cards. Remembering that at this time, personal computers were not readily available, never mind topographic maps or GPSs. The distribution maps that were created for the OHS were basically plotted manually with dots on top of topographic maps. Oh, sorry. Thank you for that, Fraser. To my knowledge, there were just 3 hard copy versions of the OHS published, and then a continual digital update until the final digital copy was released in 2000. This digital copy is no longer available online and sort of just highlights the importance of hard copies of resources such as this. Next slide. In 2008, Deb Jacobs, who I've identified here as the instigator, proposed a new reptile and amphibian atlas to coincide with the Endangered Species Act revival, Endangered ESA 2007. Again, there was a dual purpose to recruit new contributors through engaging people of varying ages, skills, and rejuvenate the collection of herp data in Eastern Ontario as the pilot. Eastern Ontario Model Forest initiated the 1-year pilot, and 2 staff were hired. Joe Crowley is project coordinator, who's on our panel tonight, as well as Oliver Reichel, who's the outreach coordinator. And I chose this picture of Oliver with a guitar because he would actually bring a guitar as a way to engage his audience in reptile and amphibian conservation. I've never seen anything like that before. One of the main goals of this project was to make data available to inform future species at risk recovery, stewardship, and research initiatives, as well as to educate the public. Based on this new initiative for recovery and conservation, a steering committee was established to help guide the atlas. And I've listed all the steering committee members that ranged from 2008 to the present, coming in and out of tenure. A few of them were even staying for the entire time of the atlas, and The three that come to mind here, Fred Schuler, Wayne Weller, and Dave Seaburn. Then at the end of the pilot in 2009, next slide, the initiative moved to Ontario wide, and with it, it moved to a provincial organization, Ontario Nature. And here it has stayed until the present day. Data collection stopped in 2019 for this initiative, but the initiative still continues to have data managers, coordinators, and managers and directors. Many of people have been involved in this effort. As the staff at Ontario Nature, as students, technicians, outreach, personnel, project initiative staff, coordinators, managers, and directors. I've highlighted all the coordinators and managers here, so you can see, for example, Emma's time here is highlighted from 2016 and continues while she was manager of the program until 2022. I think I messed up for Samara there. And so we start with Jill Crowley in 2009 and go all the way down to present day, which is Tegan coordinating and Jenna managing. Also, I wanted to give a shout out to Ann Bell, who is the director, had been the director since 2009 to 2022, and often never gets the credit she deserves for the administration, operational plans, and finances. I know I wouldn't have been able to do a lot of the things that I did at Ontario Nature without her guidance. I'd also like to give a shout out to, next slide, our data gurus. So these are often the unsung heroes behind the data, and the quality control and the reliability of the atlas depended on these people. They're often overlooked for their amazing and very important contribution. I'd also like to highlight the great, those are Ross Bushnell, Catherine Jimenez, Bradley McGinn, Mackenzie Nichols, Samara Sukumar, and Brittany Vezina. And I would also like to highlight at the bottom here, we have Alan McNaughton from the Toronto Entomological Society, who came on in 2012 as a volunteer. And he's taken the interactive range maps for the atlas and has them posted on the TEA website. For those that are interested in many different taxa, they also have an atlas and butterfly, moth and butterfly atlases there as well, all from Alan's initiative. Next slide. Again, going back to the I'm not old category, there have been a few game changers since the 1980s when the initiative started. When I talked to people that were involved in the first OHS, things that they talked about as being game changers were a standardized metric coordinate system, such as the UTM, handheld GPSs, and I would also put in their GIS systems, moving away from those hand-drawn base maps with dots, digital cameras, phones with cameras, phones with GPS and cameras, and then smartphones with cameras, GPS, apps, and immediate reporting capability. And so easy that these cutie kids here, we were teaching them, and they successfully were able to submit observations into the app with just very little guidance. Next slide. Like many things in conservation, this would not be a successful initiative if we were working in isolation. This is especially true with community science projects. But while we had a variety of staff over the 10 years of data collection, we expanded the reach of the atlas by recruiting around 50 regional coordinators and organizational champions. Some pictured here from the volunteer appreciation event that we've hosted. We also changed the vetting system of the species to be a triple check verification. With that, we needed more hands on deck. And so we recruited vetting cloud experts to ensure that species were identified correctly. And that took a lot of time and effort as well. I've been told that there's been, in the years since I've left the atlas, there has been a number of volunteers helping reach out to observers to get more correct information or clarify things and really help with the data set. So thank you to all those volunteers. And lastly, next slide. Continuing theme through history of this atlasing has been the dedication of naturalists, encouraging, educating, and inspiring the next generation of conservationists. Indeed, in this timeline, I have shown how Dr. Bleakney encouraged and inspired Dr. Cook, who later took over his role at the Canadian Museum of Nature. I started as a summer student under Mike Oldham and Don Sutherland at the NHIC, working on the OHS, and years later, and I'm not going to say how many, I became the coordinator and the manager of the atlas. Others pictured here were inspired, continue to inspire and guide conservation. Many attended a outreach session, were engaged in some way in a hobby, and then made it their career by approaching conservationists and moving it into their job. I would like to, I see Ryan's on the call there, and Catherine, and they'd just like to give a shout out to them. As we were, I would like to give a shout out to all the people on this line who continue to inspire and continue to guide conservation work, and will continue with the data gathered in this atlas. Thank you.
Thanks so much, Tanya. And now we'll welcome Joe, who's going to walk us through one of the species accounts in the herb atlas. So you know what you can expect to learn about all the species of reptiles and amphibians in the province. So take it away, Joe.
All right, thanks, Jenna. Good evening, everyone. I'll walk through what the type of information you would find, and then I've chosen a species to kind of go through and give you some examples of the type of information. The key pieces of information that we included in the species accounts are obviously the species name, the English, the French, and the Latin, a description of the appearance of the species, its biology, so its habitat use and behavior, habitat as well, the range where it's found in the province and broadly, and as well as then the atlas trends that the data we're able to look at. Next slide, yeah. So I chose the Eastern Massasauga, which won't be a surprise to most of you. This is my favorite species in the province, not just herp, but probably my favorite species. So you can see on the left-hand side there, we have the little conservation status panel. So that goes over the conservation status, the provincial under the Endangered Species Act, the federal status under the Species at Risk Act, the IUCN, International Union for Conservation and Nature status, and the NatureServe global, national, and subnational status. So it's a nice little overview of the status of the species, whether it's a species at risk or not. And then the first section is the appearance, just what the species looks like. In the case of Massasauga, it's a very beautiful snake. It's a large, it's a thick-bodied snake. It can grow to 55 to 75 centimeters in total length, although some individuals can be larger than that. I believe the record is a little bit over 1 meter long. The species has, it tends to be gray to brown, varying different amounts of gray-brown. It can even be sort of a very light golden brown, like you see in the one photo there. It can be really pretty. The species has several dark stripes running down the face, as well as a light stripe underneath. Massasaugas are a viper. So a few key characteristics of vipers include a triangular shaped head and a vertical pupil. It's the only species in Ontario that has a vertical pupil. The Massasauga is in the subfamily under vipers of pit vipers, and pit vipers have a heat-sensitive pit on each side of the face between the nostril and the eye, a little bit lower than the nostril and the eye. You can see it in a few of the photos here on the screen if you look closely. And that allows them to see a thermal image of the environment, which is a really, really neat adaptation for the species for hunting. It's warm-bodied prey, primarily small mammals, as well as just being able to see a thermal image of the environment, which obviously is very useful for an ectothermic animal, where so much of its behavior and ecology is centered around temperature. So that's pretty neat. What else do we have here? So obviously it's a rattlesnake, so it has a rattle. It's important to note that the rattle can break off. So not all individuals would always have a rattle, but they typically do. When they're first born, you can see a photo there on the bottom left of a cute little neonate that would have been just born recently. And it's got the one little button on the tail, and they add a segment every time they shed. So rattlesnakes do typically have rattles. And they have keeled scales. So they have a ridge running down the center of each scale, which gives the snake a very rough sort of texture, which is an important ID characteristic. Oh, I didn't mention in terms of the color, the pattern, it's a blotched snake, and the blotches tend to be butterfly or saddle shaped. And then it has smaller blotches, smaller alternating blotches along the side. See, we'll go to the next slide, Jenna, for the similar species. So the species that it can be confused with, there's a number of other blotchy or pattern snakes in Ontario that the Massasauga can sometimes be confused with. These include the eastern fox snake, the eastern hognose snake, the eastern milk snake, and the northern water snake. So the Massasauga has a number of unique features that no other snakes in Ontario have, including the vertical pupil, the heat-sensitive pit, and the rattle. So some really distinctive characteristics that can right away help to set it aside from the other species. Comparing it to the fox snake and the milk snake, the fox snake only has lightly keeled scales, and the milk snake has smooth scales and has a very shiny appearance. So the heavily keeled scales on the Massasauga can help differentiate it from those species as well. In addition, if you're looking at a Massasauga versus a milk snakes tend to be a very slender snake, whereas Massasaugas are very heavy-bodied. So another big difference there. The hognose snake has an upturned snout, which is a feature that helps to set it aside from the Massasauga. And the water snake, rather than having those saddle or butterfly-shaped blotches, it tends to have banding around the body. So lots of different ways, if you look closely, to differentiate it from these other species. One thing that's important to note is that some of these other species, the milk snake and fox snake in particular, will vibrate their tail sometimes if they're agitated. So sometimes people will see one of these blotchy snakes, it'll get spooked, it'll vibrate its tail, and it will be mistaken for a rattlesnake. So it's just good to know that these other snakes will also vibrate their tail if it comes into contact with some vegetation or leaves or even just the ground. It can make a rattling sort of sound that can throw you off too. So just know that you're looking for a rattle and a very distinct rattling sound if you're using that to help you identify the species. Okay, the species accounts also cover the biology of the species. So in the case of the Massasauga, they tend to be active from late April until early October. They mate from July to August, and the males move around quite a bit during the mating period. The females are able to store the sperm and then give birth the following year. So they'll gestate the young over the summer the following year and then give birth anywhere from late July until maybe early September, depending on the timing and the weather. The seasonality. The females will give birth to 9 to 19 offspring. They give live birth, so it's a viviparous species. Individuals reach maturity around 3 to 5 years of age, and the female Massasaugas will only reproduce every 2, sometimes every 3 years. So fairly slow reproduction in the species. They can live to be 17 years old. That's the oldest known record that I'm aware of. It's possible they can live older, but most individuals would not get that old. But that's sort of a maximum known lifespan. Massasaugas are a sit and wait predator. So in terms of their behavior, they spend a lot of time just sort of sitting around. They sit around in the sun and bask, and they sit and wait for their prey to come to them. I mentioned they primarily eat small mammals. So they're sit and wait predators, and they'll find rodent trails and just wait for the animals to come to them. So they spend a lot of their time just sitting around, sitting still. They're very well camouflaged. So even though they might be out in the open, or at least partially in the open, that cryptic coloration really does help them hide from predators. And while they spend quite a bit of their time sitting around, when they do move, they can move quite far. So sometimes individuals will disperse over a kilometer from their hibernation site when they're moving from their hibernation site to their summer habitat in the spring. And then again, when those males are looking for females during the mating season, they can move fairly significant distances during that time of year. So yeah, that's a little bit about their biology. So the Massasauga is venomous. I think most of us are aware of that. Rattlesnakes are venomous. Their bite can be potentially life-threatening, although there's only been a few known fatalities in Ontario. They all occurred more than 50 or 60 years ago now, and at least 2 of the instances, or the only 2 really documented instances, the individuals didn't receive any medical attention. So with our current knowledge of how to treat snake bite, as well as the the medical procedures and sort of technology and practices that are available, it's extremely unlikely to be life-threatening, but it is still serious. It's important to take it serious. That being said, bites are fairly rare. There's usually only a handful each year. The data in the atlas suggests that for the period that they looked at, or that we looked at, there was only about 2 bites per year that occurred in Ontario. It's pretty easy to avoid getting bitten by a Massasauga if you're aware they're there and you take the proper precautions. You wear boots, long pants, don't go running barefoot through great Massasauga habitat, and watch where you put your hands and feet, and you'll probably be okay. So it is venomous, but it's not something to get overly concerned about. It's just good to be aware of it. And then one of the other things that we have in the Atlas accounts, in addition to a description of the biology and all these different behaviors, is a histogram, which shows when the occurrences were reported to the Atlas and gives us a sense of when the species is active and most active, or at least when it's most observed, which often is correlated with activity. In the case of the Massasauga, you can see this histogram here, and those observations came in primarily, over the active season and peaked in August. August is when the females are gestating, and so they're a little bit more conspicuous at that time of year. It's also when the males are moving around and looking for females. So that's when we ended up getting most of the observations for that species. It can be really informative and interesting as you go species to species to look and see when those occurrences came in. Next slide, please. Actually, I'm wrong. If you can go back. Sorry, I stopped to talk about habitat. So another important piece that we have in here is the habitat of the species. So where do they live? In the case of the Massasauga, you're primarily finding Massasaugas in Ontario, where they occur in open canopy habitats that have lots of sunlight and warmth. Rattlesnakes require quite a bit of heat in order to digest their food and develop the embryos and carry out their life functions. So you find them in generally open, warm, sunny habitats like forest edges, rock barrens, meadows, all kinds of different wetlands, shorelines, basically any habitats that have a fairly open canopy and are getting sunlight, which could include small forest clearings. Forest clearings don't have to be large in order to be nice basking habitat for Massasaugas. Within those areas, though, they do require a fair bit of ground cover to provide a bit of a thermal gradient so they can move from slightly shaded to more open sunny areas, as well as to provide protection from predators. So you can see in the photos here, these are two examples of some different types of Massasauga habitat. So although we say, these are open canopy habitats, at the ground level, at that microhabitat scale where the snakes are living, you can see there's actually quite a lot going on. There's a lot of cover, there's a lot of vegetation, there's rocks, there's lots of places for them to hide. And when you have a cryptically colored snake, it can be very well camouflaged in these habitats. They also require a specialized type of basking habitat known as a gestation site. The females use these when they're developing the embryos during the summer before they give birth. And Massasaugas overwinter underground where they can get below the frost line, typically using cracks in the rock to access underground chambers or mammal burrows. In eastern Georgian Bay, they often overwinter in peatlands, but just they're getting below the ground surface. Now we're ready for the next slide, Jenna. So in terms of where the species occurs in Ontario, we have two different populations or designatable units of Massasauga. We have the Carolinian with just two very small subpopulations. One, the Ojibwe Prairie subpopulation in Windsor and LaSalle, and the other is the Wainfleet population on the Niagara Peninsula. And then the species is also found in the Great Lakes St. Lawrence region on the Bruce Peninsula and the eastern shore of Georgian Bay. So those are the two different kind of broad populations and where the species occurs in Ontario. This is the limit of the species Canadian range. It only occurs in the province of Ontario, and nationally it occurs in the northeastern US, but it has become quite rare in most of those states. And Ontario, interestingly for the species, even though we're at the northern edge of the range of a lot of these reptiles, our Massasauga populations are among the largest intact, especially obviously the Georgian Bay, in North America, along with northern Michigan. So they're very important populations for the species. Next slide, please, Jenna. So another key part of the atlas, and really where we dive into the data, is the atlas trend section. And so you can see here we have the atlas, the distribution map. And so the atlas trend sort of walks through what we found with the atlas, with all the data that was collected, where we have seen the species, where we used to see the species but don't see it anymore, and some new spots where it's popped up. So you can see from the map here, Jenna, I don't know maybe if it makes sense to go to the other map. and have a quick look at that first. So this is showing prior to the atlas, the data that we had in terms of the red being historical, yellow being recent, and the greeny blue being both recent and historical records. So then if you switch to the other map, Jenna, you can sort of see, and if you go back and forth a few times, maybe sort of how that has filled in quite a bit in terms of more recent occurrences of the species. And then you'll also notice something that isn't changing here are all those historical squares in southwestern Ontario. So if you just want to leave it on the most recent version for now, Jenna. So from that data, we can see that the species distribution has remained relatively unchanged on the northern Bruce Peninsula and the eastern shore of Georgian Bay. For the most part, we have a lot of squares there that have both historical and recent occurrences. So that means the species range has remained relatively stable in that area, as well as those two tiny little spots in southwestern Ontario, Windsor-LaSalle and Wainfleet, where we have those two Carolinian subpopulations. You'll notice a lot of the yellow squares, particularly at the northern extent of the range. Those are sites where we have seen the species over the past 20 years and didn't have historical or records prior to that. In a lot of cases, this represents increased search effort for the species, rather than it's not that the species is moving or its distribution is actually changing, but we've been looking in more places, so we're finding new records. And this trend isn't as apparent for Massasauga as it is some of the other species, but some of the species, we really got a lot of new information on the northern extent of species ranges, where we just hadn't had a lot of search effort in the past prior to the ORAA, and now we see the very large areas of yellow clusters up along the north end of the species ranges, where we're finding that they're ranging much further north than we previously knew. So that's really, really neat. You see a little bit of that here with the Massasauga. Going back to the historical squares, in southwestern Ontario, the species has been extirpated from that part of the province, due largely to the extensive habitat loss and all the other threats, the roads and the human persecution and all the things they've been facing over the decades. So those historical squares do represent extirpations of the species throughout the vast majority of that region. We also see a little cluster of historical squares on the southern part of the Bruce Peninsula and in other areas around Georgian Bay, primarily a little bit further inland from the shoreline of Georgian Bay. And with the occupancy modeling that James did, that he's going to talk about a little bit later, what we have seen with those squares is that there's a fairly low predicted occupancy of the species occurring in those historical squares, which suggests primarily due to a habitat loss, a lack of habitat right now. So it suggests that the species probably has been locally extirpated from a lot of those historical squares around the southern part of the Bruce Peninsula and southern part of eastern Georgian Bay. But something else that's really neat about the occupancy modeling that I'm sure James will talk about is that it also highlights some of the areas we might want to put in more effort looking for the species. And so we've sort of seen in Manitoulin, in the Manitoulin area and southern Sudbury district, there's a lot of areas with relatively moderate predicted occupancy of 40 to 60%, suggesting there's a reasonable chance the species might be there where we don't have occurrences. So it's sort of also this section of the atlas helps us think forward and think about where we might want to focus search effort in the future, in addition to providing information about how the species distribution may have changed in the past. So there's some really neat stuff in there in this section. Part that I skipped over, and it'll be the last thing I have here, is just in total, we have just under 13,000 records of the Massasauga in Ontario after all the efforts of this atlas and the previous atlases. With the ORAA, we ended up going from 151 to 176 squares with known occurrences of Massasauga in the province, and you sort of saw that with those two maps going back and forth there. Yeah, and I think I'll leave it there.
Great, thanks so much, Joe. That was amazing, and that's just one of many species in the ORAA. So check it out and read about your favorite species. And so I'll now welcome James to speak about a little bit more of the data and some of the research that went into the project. So you can take it away, James.
Great. Thank you. Can you hear me okay?
Yes, I can hear you fine, sorry.
Perfect, thanks. So thank you, everyone. I'm excited to share with you some results of a study that we worked on looking at how habitat loss and road density affect reptile occupancy. And so this will be a story that both looks at how threats affect species, but also a story about how we can use data submitted to the Ontario Reptile and Amphibian Atlas to learn more about their biology and also promote these species conservation. Next slide, please. So before I begin, I want to acknowledge my co-authors and collaborators in this study, including Tanya, Emma, Samara, Brittany, Ryan, and Christina, who were vital in both coming up with the ideas behind this study, but also working on the analysis and the writing and being very supportive throughout the process. Next slide, please. And so to set the stage, if we zoom back a little bit from reptiles and amphibians, we're currently in the midst of a global biodiversity crisis. And right now, biodiversity is declining faster than at any other time in human history. And the most recent global assessment report on biodiversity and ecosystem services stated very clearly that the #1 largest direct driver of biodiversity loss is changes in land and sea use. What that looks like in Ontario is expansion and intensification of agriculture, as well as resource extraction and development for industry and housing. And so if we look what that looks like on the landscape, next slide, please. We can see that especially in the southern part of Ontario, much of the habitat and the landscape has been altered from these activities. And so this area where humans have vastly changed the landscape also happens to be the place where the diversity of reptiles and amphibians is the highest. And when you have all of these activities together, you also end up with infrastructure to be able to move both goods and people from place to place. Next slide, please. And that means that this area also has a very dense road network. And so what that means is that all reptiles and amphibians likely have to interact with roads at some point in their life. They either have to cross a road as they move between habitats, or they're going to be affected by roads being close to their habitat, for example, from pollution. Next slide, please. We were interested in asking the question, what affects whether a species is present at a site? And we considered several of the largest factors, including climate, such as the temperature and precipitation, the habitat type and amount, because species occur in different habitat types, but we were most interested in understanding how these species are affected by the largest threats to their populations, including habitat loss and road density. When I say road density, I mean the total length of roads in a given site. And so we considered all these factors to understand whether a species occurs or not in an atlas square. Next slide, please. But we also knew that both in an atlas project, such as this one, but actually in any field project where you're collecting data, one of the biggest challenges is that many of these species are hard to find. And so they have what we call imperfect detection. Even when a species is present at a site, if you go out to look for it, often you won't find it, even if it's there. And so I think this picture illustrates nicely an example of this, where in the center of the picture, there's a spotted turtle that's cryptically basking under some grass. And on the right side of the picture, you can see the boot of a researcher walking by who has missed this turtle. And so we knew that we wanted to use analytic techniques that can help separate this process of not being able to detect species perfectly. Next slide, please. And so our specific objectives were to test how habitat loss and road density affect reptile occupancy, and then to be able to use our models to be able to identify new locations of reptile populations so that we can better understand trends in these populations, but also to know better where some of these species live. Next slide. And so we used all reptile observations submitted to the Ontario Reptile and Amphibian Atlas that occurred between 2009 and 2018. These observations were divided in 1,622 atlas squares, which are 10 by 10 kilometers. And these observations came from more than 5,000 unique observers. Next slide, please. We analyze our data in an occupancy model framework, which can account for variation in search effort, because some sites have lots of observers visiting them and others have very few, but also differences in habitat variables between the sites. And the model separately estimates detection probability, that's the chance of finding a species if it's present, and occupancy probability, that's the chance of a species occurring at a site given the variables there, such as the climate and habitat. We use these models to be able to predict relationships, such as how species respond to habitat loss, but also to predict locations. So we could identify places that have suitable habitat, but no records for a species. Next slide, please. And so I'll get into our results. Our first result, reptiles were less likely to occupy sites with more habitat, which is kind of an obvious statement, but something that was very apparent in our results. On the left, I'm giving the example of predictions from Blanding's turtle, and on the right, the model predictions for Eastern Massasauga rattlesnake. You can see that on the vertical axis is occupancy probability. So when the line is at the top of the graph, it means it's very likely the species is there. And when it's at the bottom, the species is very unlikely to use that site. And then on the horizontal parts of the graph, we're moving from a natural site on the left to a very agriculture and urban dominated site on the right. And although this result is somewhat obvious, being able to estimate what the shape of these curves is like for different species is very useful, so we can understand at what point species are likely to drop out with increased development. So it allows us to understand whether species can still persist in certain habitats with certain amounts of human activity. Next slide, please. So we found that habitat loss negatively affected occupancy in 19 of the 22 reptile species we were able to model. And so those slopes look different for different species, but most of them had that negative trend. Next slide, please. However, surprisingly, road density affected occupancy in only a few reptile species. So for almost all of the study species we looked at, road density did not strongly predict where species occur. Now, I want to reiterate that roads are still a threat to reptiles, but that road density, so the length of roads in a site, was not a strong predictor of whether a species occurred at a site or not. Now, there were two species that had very slightly positive effects in terms of the coefficient of road density on occupancy, and that was for snapping turtle and midland painted turtle. However, when we looked at this in more detail, we understood better that it was because these species are occurring at many sites with very high road density. So we go to the next slide. This is the confirmed and predicted occupancy for snapping turtle. You can see that across all of Southern Ontario, where there is data for turtle species, almost all of the sites have confirmed observations of snapping turtle, even those sites with the very highest road densities in our study sample. And so although the coefficient was slightly positive, it didn't actually mean that snapping turtles like to live in places where roads are dense. It more meant that they occur still in areas that have very high numbers of roads. Next slide, please. The next question we looked at answering was whether these two threats interact together. In other words, did roads amplify the negative effects of habitat loss? And simply put, no, we didn't find that. We did find a small interaction in 3 species for eastern milksnake, midland painted turtle, and spotted turtle, but overall the effects were very weak and didn't significantly affect the probability of a species occupying a square as you varied that road density. Next slide. So to summarize the results of this modeling approach, we found first, habitat loss was a very strong predictor of where species occur. Next, we found that roads were not a very good road density was not a strong predictor of where species occur. And then finally, we found that roads did not amplify the negative effects of habitat loss. Next slide, please. Although understanding how these threats affect species is a very useful exercise, we wanted to take this a step further In other words, how can these results help us conserve reptile populations or understand where these species live? One of the ways we can do this is use our model predictions to identify places where we should go out and survey for new populations. So in this hypothetical map, there are atlas squares. The green squares, the species are confirmed present. And then we used our modeled occupancy with the different colors to identify places that have highly suitable habitat, but don't yet have any records of that species. So in this example, visiting sites that are yellow have a very high probability of that species occurring there, but we don't yet have records. So they would be very good places to survey for that species. I'm going to demonstrate this more concretely with one species as an example. Next slide, please. This is the confirmed and predicted occupancy of Blanding's turtle in southern Ontario, and I'll draw your attention to an area of squares west of Ottawa. If you advance one step further, there will be an arrow, Jenna. So this is an area, a cluster of squares that are bright yellow that have very high predicted occupancy. In other words, there's highly suitable habitat there, but we don't yet have records for that species. So in terms of identifying new places to look for population of Blanding's turtles, this would be a good area to do so. Next slide. So the take-home messages that I want you to remember from my presentation. First, habitat loss is a major threat to reptiles and was very good at predicting where species occur. Next, we need to know where species persist in order to evaluate population trends and to prioritize conservation actions and surveys. Finally, I hope this example demonstrates how the Ontario Reptile Amphibian Atlas and other community science projects can be a very powerful tool to understand where species live and how they're being affected by large global threats. Next slide. Before I end, I want to thank all of the Atlas participants and supporters, whether you submitted observations, helped vet data, or supported the project in other ways. You're an important part of this project. This study that I talked about tonight is featured in the Atlas as a small chapter, but if you want to read the more detailed scientific peer-reviewed publication, you can check out the top QR code. And if you want to see our web tool, which has the predicted occupancy and the relationships between habitat loss and occupancy for all species except a few sensitive ones, you can visit the bottom QR code. Thank you very much for having me, and I look forward to answering your questions at the end of the webinar. I will pass it back to Jenna.
Thanks so much, James. That was so great. We're going to now hear a little bit about salamanders from Patrick. Like I had mentioned, there's a video that he supplied. So I'm going to get that going now and hope everything works well. And I will mention this is a shortened video. So when we send out the follow-up information from this webinar, we'll also include a link to the full video from Patrick. So if you're really interested in salamanders, please watch the whole video because it's really interesting. So I will get this going.
Proposed atlas. Salamanders the world around are amazing creatures, and I don't think I need to convince anybody online about this. Their diversity of color and pattern, form, and size is really astounding. And so it's only natural that they capture the imagination and the likes of yourselves who've submitted umpteen records of them. What I find particularly fascinating about salamanders is their role in the ecosystem. This image here shows the biomass, that is the living tissue mass in kilograms per hectare of different groups of life, large carnivores, songbirds, small mammals, herbivorous lizards, snakes, and so on. And when we scale this biomass up an entire magnitude, we can see that the humble salamander here really punches above its weight. In some of the intact forests of eastern North America, salamander biomass can reach 100 kilograms per hectare. Now, each salamander is not very big, but as a collective, they are very big and mighty. And because of this, they have this really disproportionate effect on ecosystems as predators and prey and roles in nutrient cycling. And so the humble salamander has a huge role to play in our wetlands and forests of Ontario. And I think this is really beautifully captured in a trail guide cover from Algonquin Provincial Park. And we see the minuscule wolves and bears and moose. And that's of course not to say that they're unimportant, but hulking above them are of course the enormous invertebrates, the large small mammals, the songbirds, and of course this Godzilla-like salamander. Although they're mostly out of sight and out of mind, they have this extremely large role to play in the ecosystem. And that comes in many forms. Salamanders living their life between water and land actively contribute to nutrient cycling between these environments. They're really important roles in the food web as both predators and prey. Their activity in the soil, once again, helps spread those nutrients. And their big biomass facilitates this nutrient transfer. And ultimately, they're, of course, this metric of ecosystem health and integrity like amphibians at large. And in Ontario and the Great Lakes region at large, we are right in the backyard of global salamander diversity. And so we have a lot to be proud of, but also a lot to take care of. And speaking of backyards, in your spring wanderings, you almost certainly have come across a curious gelatinous mass in the water. Now, these, of course, are the egg masses of the spotted salamander, our creature feature this evening. Maybe you've had the good fortune of not just seeing one, but many. And the spotted salamander has this curious situation of having clear egg masses, intermediate egg masses, and these very cloudy egg masses, which have been wonderfully captured and shared through observations in the ORAA over the past decade. But something that's even more curious is this. It's an algae-infiltrated egg mass of the spotted salamander. And maybe a lot of people have seen this and written it off as being unhealthy. You know, maybe these late-stage embryos aren't going to make it. Is this decay? What exactly is going on here? Well, I'm happy to report that it's nothing detrimental. In fact, it's an amazing natural phenomenon that benefits both parties in this very unusual relationship. And once again, looking into the water and seeing these greenish tinged eggs is a really unusual phenomenon, and getting up close can give you some perspective that in large part that green hue is actually coming from within the capsule of these embryos. That is, that the algae is directly associated with developing embryo, which leads to a number of questions. What exactly is going on with this algae, and why would it at all be associating with these salamanders? Well, this strange relationship has been well known for at least 130 years, and almost certainly a lot before that from indigenous persons living on the landscape. But at least to Western science, this phenomenon has been known for about 130 years, and it's garnered its fair share of attention. The relationship between the algae and the spotted salamander is called symbiosis. That is a mutually beneficial relationship or interaction between two different organisms, and two very different organisms indeed. Here we have algae, a solar-powered single-celled organism, and we have a multi-celled backbone vertebrate animal living in close association. And this particular relationship, this symbiosis, is mutually beneficial in several ways. In fact, algae, like a lot of other solar-powered life, such as plants on this globe, take the carbon dioxide that's excreted through the growth and development of the salamander, as well as nitrogenous waste, combines that with sunlight to thrive and grow. That's why throughout the developmental period of these salamanders, you see those egg masses get progressively more green. As a byproduct of the algae's growth and reproduction, it emits carbon and oxygen. Those are both vital building blocks of life for backbone organisms and a lot of organisms for that matter. So the salamander contributes the necessities for growth and reproduction that the algae needs, and the algae in turn feeds that right back to the salamander in the form of carbon and oxygen. This is the only example that we know of in backboned animals when we can get such an unusual association. The closest analog in the natural world is the symbiosis that is coral. Coral is an invertebrate animal, and it is host to different colorful algae that, again, exist in this positive cycle of sharing nutrients and thriving. And we know that, in fact, both parties benefit, especially on the salamander side of things. So the algae promotes enhanced survival of salamander offspring. And that's shown on this graph right here. For instance, this is an experimental research in which salamanders were raised in 24 hours of light, 12 hours of light and dark, or 0 light and 24 hours of dark. And in this treatment here with full sunlight, which promoted maximum algae growth, you can see that the embryo hatch rate skyrockets. Not only do these embryos develop faster, but by the end of development, they end up achieving higher hatch success or higher survival. So this algae not only promotes faster development, but larger sizes at hatching, higher success of survival at hatching, and it tends to synchronize hatching, leading to most individuals ultimately escaping their egg mass right around the same time, which can be very beneficial to avoiding predators. And so this really speaks to that symbiotic relationship that the salamander helps the algae grow, the algae helps the salamander grow, and contributes in several other ways as well. But this situation gets even stranger. Not only has there been a lot of research poured into this relationship about salamander embryos and the relationship with algae over the past decades, but within the past several years, there's been new research to show that not only does the algae live in association with salamander embryos in their egg capsule, but some algae actually lives within terrestrial juveniles and adults of the spotted salamander. Now, this is really perplexing because once the salamanders move to land, of course, they only return about once a year to breed in the water. And algae, well, it normally lives in water. So how does it manage to infiltrate the tissue and live within the body of a spotted salamander? Not only how does it manage to live inside the tissue, but of course, these salamanders spend most of their life underground. And so what does a solar-powered organism like algae, what is it doing living inside the body of an animal that spends almost its entire existence in darkness underground and also only active at night? Well, the amazing thing about nature is it's not giving up its secrets easily, and we really don't know. What we do know about this algae that lives within the reproductive tract of salamanders is that it can contribute in important ways to carbon fixation, that is continued growth, but it also raises a lot more questions. For example, the algae has not been successfully documented free living in natural ponds. So does it really only live and cycle through the bodies of these salamanders, getting deposited into eggs? There's a whole host of strange and wonderful questions that are waiting to be explored. Composed atlas.
Okay, great. That was wonderful. Like I said, that's just a little bit of a longer video. So please watch the full video once you get the link. And now I'm going to turn it over to Emma now to discuss some impacts and next steps.
Thanks, Jenna. And thanks to everyone for joining tonight to celebrate this milestone for the Reptile and Amphibian Atlas. I'm going to, as Jenna mentioned, I'm going to be walking you through the impact and next steps section of the publication. And of course, this project has supported reptile and amphibian conservation efforts in Ontario in several important ways. And through this section, I'm really going to focus on highlighting 3 key impacts and then briefly speak to future directions. Next slide. So first and foremost, the collection and compilation of species occurrence data has greatly improved our knowledge of reptile and amphibian species distributions province-wide. At the onset of the ORAA, despite successful data collection efforts associated with the Ontario Herpetofaunal Summary,
there was still a noticeable lack of detailed, up-to-date occurrence data for several species in many parts of Ontario. And this presented considerable barriers to species management and conservation efforts. And so there really was this collective recognition that there was a need for surveys and inventories to increase knowledge of species distributions. And this was included as a recommendation in the actions for recovery strategies of almost every amphibian and reptile species at risk. And the ORAA has really made considerable strides in terms of filling these knowledge gaps and addressing these recovery actions for Ontario's at-risk reptiles and amphibians. And the species account section of the publication provides in-depth details for all of Ontario's reptiles and amphibians, including the species occurrence data, which Joe has talked about in his example with the Massasauga rattlesnake. Here I'm also highlighting another example with the American toad. A total of 22,187 observations of this species was added to the ORAA across just a little over 2,100 grid squares for the province. And for each species accounts, you'll see these distribution maps, as well as a discussion around data trends for each species. For the frogs and toads, you'll also see the number of visual records versus calling records by month, which you can see in the bottom left-hand corner of the slide. And I just wanted to point out as well with the species occurrence data collected through the ORAA that it plays really a critical role in informing species status assessments and designations by CSIWIC, as well as CSARO. And both of these committees determine the designated status of species under the Species at Risk Act and the Endangered Species Act. So that data has played a central role in those assessments and designations as well. Next slide. The second impact that I wanted to highlight is public education and engagement. So the Atlas has increased the public's understanding of and appreciation for reptiles and amphibians through education and outreach, engaging the public in reptile and amphibian stewardship and community science. And over the 10-year lifespan of this project, the ORAA has had over 12,000 contributors, and an additional 4,000 people submitted observations through the OHS from 1984 to 1995. Additionally, approximately 1 million people were engaged in the ORAA from 2018 to 2022. And this is largely through online engagement. And as sort of was highlighted by Jenna earlier in her presentation, is that there are an additional 27,000 individuals that were engaged over 260 events. So those would be, through field courses, training workshops, as well as stewardship and outreach events. So this is one of the, second main impacts that the ORAA has had. Next slide. And the third impact that I wanted to highlight, and is highlighted in the publication, is that the ORAA has really had a major impact in terms of collaboration and research. It has strengthened and supported key conservation and research initiatives by facilitating collaboration among herpetologists and conservation professionals, and providing a foundation for developing and strengthening conservation partnerships with private landowners, municipalities, Indigenous partners, and other land-based sectors, such as woodlot owners and agricultural practitioners. I don't think that it can be overstated just how fortunate this project has been to have a dedicated and passionate group of volunteers and staff who have contributed greatly to its success. And from the ORA's foundation that's so strongly rooted in collaboration and partnerships, It has supported numerous research projects. And James's presentation, as well as Patrick's, have highlighted how this data can be used and applied in conservation and research. And there are more examples of specific examples of research that are and conservation applications that are highlighted under the impacts and next steps section. So I would encourage you to check that out if you want to learn more. And next slide, Jenna. So looking ahead, after a very productive and sustained period of data collection from 2009 to 2019 of community scientists submitting observations to the ORAA, I think now at this landmark is a good time to look forward and identify opportunities for future research as well as stewardship needs. Next slide. And so some examples of future research that I'll highlight. So of course, this data set has the potential to answer a wide range of research questions related to species phenology, occupancy and detectability, regional and wide range distributional patterns, or the effects of climate change on northern range limits and species biology more generally. The occupancy model and maps developed by James and highlighted in his presentation have identified atlas squares that are likely to support reptile populations, but have had no observations since 2009 when the ORAA began. So these maps can be used to prioritize future search efforts by identifying atlas squares where there's the highest probability of reporting unknown populations. And that's represented in the two maps shown here on this slide, where you have squares in teal and orange that are showing sites that have where there are likely to be populations of snakes and turtles, but haven't been reported. And within each species account, there is further discussion around priority areas for future sampling and discussion around uncertainties that really lend themselves well to identifying future research opportunities in terms of range gaps, northern range boundaries, and zones of subspecies intergradation. I also want to highlight that while there's a lot of new data that was collected across central and southern Ontario as part of the ORAA, northern Ontario remains one of the most undersurveyed parts of the province for both reptiles and amphibians. And in fact, the northern range limit of most species still remains uncertain, which really decreases our ability to track changing distributions. And this is particularly true for species that are found only in northern Ontario, including the red-sided garter snake, western painted turtle, the boreal chorus frog, and central newt. And so there are ongoing opportunities there to increase our knowledge across the north. And then lastly, one of the key differences between the ORAA and the OHS was the ORA's ability to archive large amounts of digital information, which Tanya talked about in her presentation, kind of walking us through the history of how data collection has changed significantly over the years for atlases. And so with many of the observations that were submitted to the ORAA, we have photos and audio and video recordings associated with these sightings. And this digital information has huge potential for answering research questions. For example, researchers could look at photographs to document and describe morphological variation or color polymorphs, or videos to look at unique behaviors. So certainly there's no shortage of interesting questions to be answered with this data set. And next slide. Lastly, next steps in terms of community science projects. Of course, there's considerable value in continuing to submit observations, even of common species in places where they've already been reported. And although data collection for the ORAA has come to a close, there are still many ongoing projects collecting data on reptiles and amphibian occurrence. And so we don't have time to talk about all those projects in this presentation, but under the stewardship section of the publication, there is a list of these projects, and I would encourage you to check that out and look at ways that you can continue to stay involved. And biodiversity atlases such as the OHS and ORAA provide a snapshot of species distributions at a large geographic scale. And these efforts provide an important baseline for additional sampling. And while the ORAA has improved our understanding of reptile and amphibian distributions across Ontario, atlas occurrence data cannot necessarily be used to gauge abundance and is limited in its utility for detecting changes in abundance or population trends. And really, to monitor abundance requires standardized survey and sampling protocols, such as those that are incorporated into Ontario Nature's long-term monitoring protocol for snakes. So continuing to support these types of monitoring and survey efforts to detect changes in abundance will continue to be important over time. And that hopefully gives you a snapshot of some of the key information presented in the next impact and next steps section of the publication. And I hope you enjoy reading it, and I'll hand it back to Jenna, who will talk a little bit more about how you can stay involved.
Thanks so much, Emma. That was great. I'm going to just quickly run through some important links that I hope you all check out. Leslie's going to pop a bunch of these links into the chat as we go here for the last few minutes tonight, but I'll wrap up quickly so that we can get a good 5 minutes of questions in at least. So ways that you can stay involved with the project and continue. One, of course, please continue to report your sightings of reptiles and amphibians to ideally the Herps of Ontario iNaturalist group, and or also for species at risk, the NHIC project. You can report direct to NHIC through their website, or they have an iNat project as well. You can keep the conversation going by joining the ORAA Facebook page, which is nearing 10,000 members. So it'd be really exciting to cross that threshold. It's a great place to get support for species ID or just talk all things reptiles and amphibian conservation and be connected with a lot of people in the community. You can access the online ORAA report, what we've been talking about tonight. It's completely freely available online. So visit Ontario Nature's website and check out the report. Hopefully it's something you can keep referencing and going back to. And there's also a great story map that goes along with it that highlights some of the key findings of the atlas. And also the Ontario Nature maintains field guide ID pages on the website, which has even more information about reptiles and amphibians in the province if you can't get enough and you need to find some more. I want to just highlight that we are also doing a limited pre-sale of the atlas. So stay tuned on the email to follow this event. You'll find a link to be able to purchase the hard copies in the future, and we're just doing one print run, so get them while the getting's good and make sure you get your copy when you need it. Sorry, do you have something to say, John?
Sorry to interject. Just hoping we can just run a quick survey. I just want to gauge interest in purchasing a hard copy. So we're sort of basing this on demand. So I'm just going to launch a quick survey here. Sorry to interrupt, Jenna, but
yeah, no, that's great. Yeah, so the copies will be available for $110, all included. That's taxes and shipping and everything. And yeah, we're trying to figure out how many to print. So let us know if you think you're going to be getting a copy here. And As well, you can always email the atlas@ontarionature.org email for anything Atlas related. If you have data requests or other inquiries related to the project, don't hesitate to reach out. And while the poll's still going, I'll just keep talking while the poll's still going. In what we have coming up next is definitely keep an eye out. We're going to be doing some outreach events in the spring to hopefully be distributing printed copies, but also just keep the conversation going about reptiles and amphibians and keep the engagement going. And many people have asked, we are planning on relaunching the Reptile and Amphibian ID quizzes. So that's to come. We're actively working on it and they should be ready by spring. So we're excited about it. Great, lots of interest in hard copies. That's wonderful. And also the last thing I'll say before we turn it over for a few minutes of questions is please also help us by completing the feedback form tonight. There'll be a link in the chat and also a link in the emails that you receive after the event. This event was funded by TD Friends of the Environment, so we love to report back to them with great highlights about your experience and also just asking some important questions about your current and future engagement with reptile and amphibian conservation. So thanks in advance for completing that. And I'll just end by thanking, of course, all of the very many funders who contributed to this project over the year. There's just too many to mention. And there's also quite a few donors in the last stretch here, the last few months that gave, and whose names aren't listed here, but we're so appreciative of. So thank you, everyone, and I will stop sharing and turn it over to Leslie to ask us a few questions with the last minutes left.
Thanks, Jenna. Thanks all of the panelists. That was super interesting. Do have a few questions, and just to let everyone know, if you have questions that you think of later or don't get answered tonight, then please do feel free to email us and we will try to get those questions answered for you later. So very quickly, I'm just going to go through 3 questions. I think we answered most of them throughout. 2 of them are for Joe about the Massasauga rattler. So Joe, quickly, is it possible that someone had seen a Massasauga in Hamilton? And also, can you say how climate change might be affecting the Massasauga rattler?
Yeah, so it's not possible to see a Massasauga in Hamilton that would be part of a naturally occurring population. We know with high confidence that at this point, Massasaugas are limited to those 2 sites I mentioned in southwestern Ontario, in Windsor-LaSalle and on the Niagara Peninsula in Wainfleet. They don't occur anywhere near the Hamilton area. That being said, it's possible that someone illegally captured a rattlesnake and released it in an urban center. It's possible that someone's pet escaped. So I'll never say it's impossible to see it, but you wouldn't have seen a naturally occurring individual in that area. In terms of climate change, there's obviously so much we don't know about how climate change is affecting these species. Certainly, warmer weather may result in a lot of our reptile amphibian species potentially being able to, over the course of a long period of time, move their ranges north. But there's a lot of other impacts. For example, with Massasauga, one thing we've been seeing in Ontario is that we end up sometimes with an increased severity and intensity of storms and weather events and unusual weather in the spring that can sometimes result in, or in the fall, that can result in a lot of freeze thaw cycles and flooding of hibernacula. So we did have a number of years ago a large mortality event in eastern Georgian Bay where because of a lot of warming temperatures and then cold temperatures and then warm again and then cold, we saw a lot of flooding of Massasauga hibernacula after the snakes were hibernating, and that resulted in very large, widespread mortality of the species. So sometimes we get those sorts of effects and impacts that can be sometimes tricky to detect, and we only were aware of that instance because of some groups that were monitoring the species in the area. So yeah, certainly climate change affects them in a number of ways. Tanya also did put some information in there about a recent publication as well on how climate change might affect the species.
Great, thank you, Joe, and thank you for those questions. The next question I think is for James. Could increased road density increase observers' opportunity to make observations of reptiles, resulting in the masking of the negative impacts of road density on reptile survival? I won't try to say that too many times fast, so you got it.
Yeah, first, Pamela, thank you for the question. That's an excellent idea, excellent question, and one that tormented us for quite a bit of time as we were talking about it. I didn't go through all the details of our modeling, but we did actually include the density of roads as an effect on detection probability, because we had the same thought as you, that roads are a way that people often observe species more easily. And so places with more roads are more likely to have more records and more access. So we did try to control for that. Now, whether it adequately did completely or not, then that's difficult to say, but we did estimate that effect. So excellent question. I encourage you to read more in the Atlas publication if you want to see the different ways we controlled for those variables. Thanks.
Thank you, James, and thank you for the question. Just one more, there was a question about where the link to the salamander video is. That will be in the follow-up email that you'll get in a couple of days after this webinar. So that'll be in there, and all of the links that we posted in the chat will also be in that follow-up email, as well as the recording of the actual presentation. So that's all coming, and it will all be available to you again. So don't stress if you miss something here today. So with that, I will again thank Jenna, thank the panelists, and thank all of the many people that contributed to the Atlas. And thank you all for coming here tonight. That's the end of our webinar.